Good morning and welcome to the Nusano office hours on Radiopharmaceuticals. At this time, all attendees are in a listen-only mode. A question and answer session will follow the formal discussion. If you'd like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player or by emailing your questions to questions@lifesciadvisors.com. As a reminder, this call is being recorded and a replay will be made available on the Nusano website following the conclusion of the event. I'd now like to turn the call over to Chris Lowe, Chief Executive Officer at Nusano. Please go ahead, Chris.
Thank you, Tara. Good morning, and thank you everyone for joining today's event. My name is Chris Lowe. I'm the CEO at Nusano. Today's event is a continuation of our series focusing on the role of isotopes in the next generation of oncology care. I'm really looking forward to listening to today's event. It's my pleasure to introduce our host, Dr. Sarah Schouten. Sarah has an impressive career which began in academia and quickly grew into sell-side equity research and executive roles in private industry. Sarah, thank you for leading us today. The floor is yours.
Thanks so much, Chris, for that kind introduction, and thank you for inviting me to host this event. This is an area that is not only exciting and interesting, but near and dear to my heart. So we have two prestigious guest experts that we're lucky to have a conversation with today and a full agenda. But first, I would like to add another facet of my experience to share with all of you. So many of us have been touched by cancer in our lives and have our own personal stories. And myself, last summer, I was diagnosed with stage 4 metastatic breast cancer. And I was very fortunate to have been given access to the world-class healthcare and the latest evidence-based treatment. And because of this has allowed me to not only survive, but thrive. And I would like to use my own story to serve as a living testament to everyone here on this call that we're all working towards the same thing, right? Bringing treatments, better treatments to patients. And I would like to personally thank everyone for working hard towards this goal, for better treatments for patients. And with that, let's get to work, right? I want to introduce first, our first one speaker is Dr. Suzanne Lapi, and she is Vice Chair of Translational Research at the Department of Radiology at the University of Alabama at Birmingham, where she's a tenured professor of radiology and chemistry. She's also an Emmett O'Neill Professor at the O'Neill Comprehensive Cancer Center, and her research interests are in the development and translation of new PET radionuclides and molecular imaging agents. She has over 150 publications and is PI on active research grants from the National Institutes of Health and Department of Energy, as well as industry partners. In addition to her intellectual expertise, she's also on the front lines as a cyclotron facility director, producing and supplying medical isotopes. She oversees production of PET radionuclides and imaging radiopharmaceuticals for preclinical research and clinical trials. Her group holds over 15 approved INDs and supplies copper-64, zirconium-89, manganese-52, and other isotopes to groups not only US, but also around the world. And then if that isn't enough, she's also just received the Michael Welsh Award from the Society of Nuclear Medicine and Molecular Imaging at its recent 2024 annual meeting that was held earlier this month in Toronto. The Welch Award honors individuals who've made outstanding contributions to the field of radiopharmaceutical sciences through mentoring students, postdoctoral fellowships, junior faculty, and community service in the field of radiopharmaceutical chemistry and molecular imaging. So congratulations on the award and welcome and thank you. We also have Kemp Dolliver with us today. Kemp is Director of Research and a Senior Analyst at Brookline Capital Markets. Kemp has an extensive healthcare experience across multiple sell-side and buy-side roles in the US and Asia. His career includes time as an analyst and portfolio manager at Religare Capital Markets, Avondale Partners, Cowen, Aetna, and Bank of America. Among his many impressive achievements, the Wall Street Journal has also recognized him 4 times for his stock picking abilities in its Best on the Street survey. Kemp received his BS in Commerce from McIntyre School of Commerce, where he currently serves on the advisory board, and he had MBA from the Darden School of Business at the University of Virginia. And finally, most relevant to our discussion today, he's the author of Radiopharma Roundup, which is a definitive analysis for all things radiopharmaceuticals for Brookline clients. So welcome, Kemp. So I guess we'll just dive into it here. Let's stop right at the top and talk about specifically what is a radiopharmaceutical? How do we define it? Because we know the known anti-tumor effects of ionizing radiation can be combined with the specificity of cancer cell targeting. So this is, many exciting combinations of isotopes and targets. So let's get to it. How do we make a radiopharmaceutical? What is that? Suzanne.
Sure. So thank you so much for having me here today. So this is my favorite topic. So I'm always excited to talk about this. So a radiopharmaceutical you can think of as a drug that's tagged with a radioisotope. And so the drug determines where the complex will go in the body, how it's excreted, and how it can target the tumor or other disease state that we're looking at. And the radioisotope that we're attaching to the drug determines whether it can be used for imaging or therapeutic properties. And so, I know we'll get into this a little bit later, but a real sort of emerging area is the use of theranostics, where you can use the same drug that's labeled with a radioisotope for imaging. We do the imaging study and we can select patients that may benefit from the same drug that's labeled with a therapeutic isotope. So really sort of using these strategies for personalized medicine.
And when we talk about radioisotopes, and we know that this is a tried and true MOI for destroying cancer cells and shrinking tumors. So what specific properties, maybe we should talk about of a radioisotope? There's different emissions, and maybe we can touch on what each of these emissions are and how they can differentially affect tumors and cancer cells.
Absolutely. So radioisotopes that can be used for imaging agents are usually in two categories. They emit photons or positrons. And so photons leave the body, and then we can use a camera or a scanner to really determine the location of those isotopes. Positron emission tomography uses isotopes that emit positrons. In the body, the positron collides with an electron, and that gives rise to two photons that leave the body, which again can be detected by a scanner that's around our patient. Therapeutic isotopes have emissions that distribute a very localized radiation dose. So instead of those gamma rays that leave the body and can be detected, that localized radiation is deposited in the cancer cell or in a very localized area of a cluster of cancer cells. And so we can use beta particles, which are essentially electrons, to target radiation to cancer cells. We can also use newer generation of radioisotopes that include alpha emitters or Auger electron emitters that emit radiation in a very, very localized spot to really deliver very targeted treatment to cancers.
Sure. And so current approved therapies right now for radiotherapeutics are mainly, they're all approved are beta, right? So we don't have any alphas yet. So can we talk a little bit more about beta, maybe the reach or characteristics specifically of beta relative to alpha, as far as what targets, target size, I guess is what I'm getting at here.
Sure. So the most commonly used are sort of in this sort of theranostic approach is lutetium-177, which is a beta emitting radioisotope that emits electrons that are a few millimeters in the body. And so these are very useful for targeting clusters of cells. And the way that I sort of in a similar fashion to what I mentioned before is we can use targeting compounds to specifically take this lutetium-177 radionuclide specifically to the sites of cancer and distribute that very localized radiation dose. I do want to note there is an approved alpha emitter. There's radium-223. And so I just want to mention that they're not all the approved drugs are beta emitters. And we're also seeing very promising studies come out of clinical trials with alphas these days as well.
So just transitioning to alpha. So why do you think alpha has that beta doesn't have? So let's talk about alpha emission and how that differs both on physics level, but also how that translates biologically.
Yeah, it's interesting. So I think that so beta particles will go, sort of a very short path length in tissue. Because they're distributing their energy over that short path length, you think about sort of the density of ionizing radiation, or how much energy they're depositing per unit length is going to be lower than an alpha particle that only goes a very, very short path length. So alpha particles can be thought of kind of like, a bowling ball versus, a basketball. So it's like a very, very dense ionization tract. It delivers very heavy amounts of energy in a very, very small location. I think one thing to think about is that the tools that we have to develop radiopharmaceuticals with either alpha particles or beta particles are quite frequently using elements that are different. And so the chemistry is also different between a lot of those radioisotopes. And so we need to think about how best to conjugate those compounds to the targeted radiopharmaceuticals as well.
Sure. So when you mentioned chemistry, which is something definitely I wanted to go into. So when we think about using different types of isotopes, right? They're essentially, they do have different chemistries, and there's different considerations on how to attach them. So maybe it's not always so simple as you have a molecular targeting agent and you can easily just switch out isotopes, right? You need other chemical considerations like chelators or linkers, right? So with the current chelators that we have now, I mean, we could talk about what a chelator is and what is available, and maybe what kind of advances that we need to make sure that we are able to use a wider variety of isotopes in a clinical setting.
Absolutely. So you can think of kind of your drug or your targeting molecule that's homing to the cancer cell. And the chelator is basically, you can think of it as a claw that holds on to the radioisotope and makes sure that the radioisotope stays with that targeting drug. The chemistry of the different isotopes, so we mentioned lutetium-177, and then let's say we have an emerging alpha emitter, this is actinium-225. The chemistry of those isotopes are going to require different chelators. So we want to make sure that we have chelators that are going to be very stable once we inject them in people, because if the chelator lets go of its radioisotope, the radioisotope is no longer sticking with the targeting vector and homing to the cancer cell, and which is what our goal is. The different chemistries need different chelators, but also an important point to make is that even if you have the same chelator, and you can have the same chelator for different isotopes, you may be changing slightly but significantly the way that the targeting vector behaves in the person. And so let's say if I have a compound that is used with lutetium-177, and then I change that to, let's say, lead-212 or another radioisotope, that change in charge and change in structure of the chelator leads to changes in the way that the targeting vector behaves. And so we need to think very carefully about the chemistry, and that's why a lot of these preclinical studies or early phase studies to really understand how the agent targets the tumor and how the agent is cleared from the body needs to be carefully understood with all of these combinations with different chelators and different isotopes.
Sure. And I think there's also downstream considerations, because a lot of these isotopes are radioactive and they emit, but then they change to another isotope with different properties. And we would want to make sure the chelator would also retain all of these decay products as well, to keep them, in the neighborhood of where we want to be molecularly, and not a chelator that would release these decay daughter products. So with that said, like when we're choosing which isotope to use, like right now, lutetium is a big one that lots of people are using. Do you believe that why do you believe this isotope is the first one that's really been widespread use and gained the most interest commercially to this
So I
point?
think the chemistry is very well developed, and so we know how to make stable chelators. that incorporate lutetium-177. And there's also excellent targeting vectors that have been coupled with the chelators that have shown a lot of promise in, obviously in clinical trials and now in standard of care. So it's really moving forward, and there's just been so much data to support the safe and effective use of these compounds. And so I think that's kind of paved the way for lutetium-177. The other thing is that the production of lutetium-177 is very well known. And so and I use this as a field, our ability to make large quantities of lutetium-177 and really get lutetium-177 into the clinics has really helped the advancement of these types of technologies.
Okay, so but the development of these new chelators and linkers would potentially provide another commercial opportunity as assets for other radiopharmaceuticals in development or to come in the future. So the chelators that we have now, what isotopes do they work with, besides lutetium? And what ones do you see as a need to get them really into the clinic?
Yeah, so I think there's a lot of very nice chelators for lutetium and for gallium, which is the imaging agent that's quite frequently paired with lutetium. We're seeing really great chelators now for copper that have showed a lot of stability. But with some of the newer isotopes, such as actinium and maybe some of the newer alpha emitting isotopes, we're still seeing areas where the work needs to be developed. I think another important consideration is that even if you have a chelator that's very stable for the therapeutic isotope, really understanding how to make diagnostic radiopharmaceuticals that can help stratify which patients may be responding to the therapy or how to pick which patients should get the therapy may still be an area of development. So really making sure that your imaging agent that you're using is really reflective of the biodistribution of the targeting agent so that you can help understand, what the tumor uptake will mean for that patient's efficacy.
Right. So yeah, so you had mentioned this earlier, set up for this is that what you're talking about is these theranostic pairs. And as we grow the library of clinically and commercially available isotopes and linkers and chelators, we can just have this whole library available to us where you can really start to personalize and fine-tune radiotherapeutics. So what kind of considerations are we having when we're talking about these theranostic pairs, right? So is it so simple as you can just switch out the isotope and,
Yeah, so it actually requires very careful consideration. And so, switching out the different isotope, it really depends on the targeting vector and how that targeting vector localizes to the cancer. And so for each targeting vector, or each asset, as you put it, careful consideration will need to be taken about which chelator and which isotope. And we've definitely seen things where one isotope works well for one type of targeting vector, maybe it's a peptide or a small molecule, but another isotope will be warranted for, a protein-based construct or another type of targeting vector. And so I think your point about the toolbox and having a toolbox where we can really look at customized transition metal isotopes or F block elements and how that and the customized chelators may be really something that we look at for new targeting vectors. And I think that needs to be really considered early on in the radiopharmaceutical development.
Sure. And as far as regulatory pathways, I mean, is there an advantage, do you think, to doing these theranostic pairs? So perhaps, you can get these new vectors into humans, maybe in a quicker fashion on a diagnostic pathway. And then that enhances your ability to bring this to patients, on the theranostic side.
Yeah, so the regulatory sort of constraints for early phase clinical trials for diagnostics are are going to be a little bit more straightforward and easier to achieve, I think. And so then you could also use the imaging trials to really understand how you would use the therapeutic and understand how you would design a therapeutic trial. I think what we're seeing now too, which is really exciting, is using the imaging agents to select patients that will benefit from the therapy. And I think by doing that, then you can really have a much better response rate, and you probably can use fewer patients in these clinical trials to illustrate efficacy of the targeted radiotherapeutic. I also think that the idea of using a very similar or identical targeted imaging agent to select which patients are likely to respond to the targeted therapy is something that's incredibly attractive to both physicians that are involved in these trials or in these standard of care techniques, and also to patients. Like this idea that we're going to image you and really see that the drug is taken up in your tumor and that you're likely to respond to this type of new drug is just, I mean, as a patient, it's really attractive.
Certainly. And it's also downstream from that after, treatment starts, it's a good way you can monitor, treatment effect, the effectiveness of the treatment. And, if there's, the treatment is still shrinking the tumors, you can see it much more easily with these diagnostic pairs. So treatment response provides another opportunity. So it takes something like maybe diagnostics that I feel like in the growing interest in radiopharmaceuticals, diagnostics really has a big supporting role and multiple potential uses throughout, diagnosis and therapy and treatment monitoring, and even down the road monitoring for recurrence of disease in patients. So there's a lot of opportunities beyond just the therapeutics as far as market opportunity. And maybe this is something we haven't talked too much about. Kemp, you've been a little quiet. Maybe this is a little bit more in your realm, but how do you see the market opportunity maybe for the radiotherapeutics versus diagnostics and currently in
Right. Yeah, so what we've seen so far is with therapeutics, the typical treatment regimen is going to be 4 to 6 doses, and the price is around $50,000. So the math, $200,000-$300,000 per patient, roughly. With diagnostics, the price points are about 10% of that. So right now about $5,000. But you will probably scan that patient multiple times during their course of treatment. I don't think you'll necessarily do it 10 times and get up to the equivalent of one therapeutic dose, but you're screening patients broadly, and for some of the newer imaging agents, particularly for prostate cancer, at least one of them's on track for about $1 billion in revenue this year.
That's interesting. So as far as what's in the pipeline, though, let's switch back, to talking about the different types of isotopes. And again, lutetium is the largest, certainly, especially more advanced as far as market share. But where do you see the future going as far as some of these new and up and coming isotopes for both therapeutic and diagnostic applications?
So for beta emitters, terbium-161 is certainly getting a lot of attention. I definitely see more work being done in that area. And then with the alpha emitters, there's sort of the 3 biggest ones, which are astatine-211, actinium-225, and lead-212, I would say, are sort of the biggest ones in the field where there's exciting clinical trials going on. I think sort of coupling isotopes that can be used to make diagnostic analogs of those are very important. I think gallium-68, which is currently widely used, will continue to be sort of a major player. But I also think that longer-lived isotopes, so we can really look at how the drug migrates for longer periods of time in the body, will also be important. And so maybe scandium 43 and 47 or might be more interesting as we move forward. I think there's also a real interest in identically paired isotopes. So copper 67 is a beta emitter that's also in clinical trials, showing some really exciting results. And the nice thing about copper-67 is that the diagnostic isotope is copper-64. And so you have a true identically matched therapeutic and diagnostic pair. So you can make radiopharmaceuticals that are identical. The same thing can happen with lead-212, which is an alpha emitter, with lead-203, which is a SPECT isotope, where you can make identically matched paired radiopharmaceuticals. And I think that allows us for a really accurate understanding of how the radiotherapeutic moves around in the body, and even might allow for, so tailored dosimetry and tailored administration, and kind of tweaking the dose so that the patient best responds.
Yeah, that sounds good. So let's see now. We're talking a lot about isotopes. I want to back up a little bit and talk about where do these isotopes come from, right? So radioisotopes need to be produced now, just to get our nomenclature right. We have a lot. Maybe let me just say this. Why don't you describe what the supply chain is from starting material to getting it to the patient, right? What steps does it require? Because it's a little bit different than what we think of with traditional, pharmaceuticals. So who wants to take a stab?
Sure. So isotopes can be produced in a cyclotron, like at our facility. We irradiate stable targets with a proton beam. We induce a nuclear reaction, and we make an isotope in that target, and then we have to process the target to get the isotope out. Similarly, we can make isotopes in a reactor where we're irradiating targets with neutrons and inducing nuclear reactions that way, or inducing fission products. And then more recently, people have also made use of photonuclear reactions where we're irradiating targets with gamma rays. In all of these cases, we're irradiating some kind of stable material, and then we're making a radioactive material. And in most cases, the radiopharmaceutical may not be made at the same site as the radioisotope is generated. So we're shipping these things around. They have half-lives, on the order of hours to days. And so there is quite a, logistical challenges. And I think that's something that companies in this space are having to deal with. And it's a little bit, as you mentioned, trickier than conventional pharmacies. And so I don't know if Kemp has some thoughts on that.
Another consideration is where you can get the predecessor isotope. So for instance, for actinium-225, the best source is the Department of Energy's thorium-228 that are called the thorium-228 cows. And, that's unused nuclear, the unused nuclear warhead material. And so, so the other isotopes all have their source material, some of which is readily available, other cases not. I think terbium, there's a high reliance on supply potentially just from Russia. So, there's a lot of work going on in the background with regard to increasing access to isotopes. And again, depending on the particular isotope.
Sure, and current production methods don't necessarily account for this big scallop and explosion that'll be required to do multiple, regulatory cleared treatments and diagnostics for patients. So, I mean, as far as production capacity, it seems that we also have to have some definitely some advances to get up to that level of production. And we've seen, recently there's been several companies that have made announcements related to production. So maybe you can talk about a little bit about this, how we've seen in, some developers and
Yep.
even commercially available issues with production.
Right. So, there are a number of different angles here, and let's just first, I guess, some good, bad, and the ugly, and maybe not necessarily in that order, but Raise Bio, which was just acquired by Bristol Myers Squibb, their Action 1 trial had to pause commencing the phase 3 portion because one of their two supply, I think they have two suppliers of actinium, was not making a delivery. And we have seen some interruptions in supply in development of lutetium in the past, but those are short-lived. And I think as long as these bumps in the road are short-lived, the industry will advance. I think when they become chronic, we have a larger concern. So that's angle one. The other consideration is see the industry going toward using short half-life isotopes for therapeutic use. Right now they're mainly diagnostic use. So there are manufacturing requirements around that require localized or regional manufacturing, and those networks have to be in place before you achieve commercialization. To some extent, you might be able to rely on the radiopharmacy network, and the radiopharmacy networks almost all of them are expanding and have announced expansion plans over the course of the last year. And then there's just the development of a contract manufacturing network to support small companies in taking small amounts of isotopes and making clinical supply.
But certainly we can see with, supply chain, if constrained to less isotopes, that's another benefit, not only, clinically and chemistry wise, but just having a broad array of different isotopes, which would have different starting materials potentially, would also free up and potentially prevent these kind of bottlenecks that you could see if everybody is going after the same isotopes. So there's a lot that needs to be done to get it up to scale, but certainly, there's solutions that, the demand is there. There's solutions that are being made to get this scale up and to get this logistics of getting these short half-lives. So
maybe I could just add to that, because I think the development of all these different ways to produce isotopes and produce radiopharmaceuticals is absolutely essential to the movement of these exciting things moving forward. I think that goes for industry sites as well as academic sites and hospitals that are working to move the clinical trials and standard of care forward, and partnerships between all of these things. Closely related to this, I just want to bring up is a real workforce need. Everybody's looking for new radiochemists, new radiopharmacists, new people that understand how to work with these isotopes, how to make new radiopharmaceuticals, how to do the purification, how to do the research to move these things forward. And so I think one of the things that we're seeing is a real need for expanded training programs to develop people that can work in this area. So I just want to make a little plug for that, because I think we're starting to see a strain in the number of people that we can train and get out there to move this forward.
Yeah, and you're also seeing KOLs move around between institutions.
Yeah, that's certainly a consideration that at the patient end, that's probably the most important, right? Once we make these drugs, to make sure we have the proper expertise to get them into the patient. So certainly an important thing to build up in the infrastructure of radiopharmaceuticals. So maybe at this point, I think maybe I want to talk about targets a little bit more. So we talked a lot about the isotopes and different types of isotopes and production methods, but what about targets? So what targets are optimal for radiotherapeutics as opposed to conventional methods that we're doing now? Where do you see the greatest opportunity beyond prostate, which is probably the most largest market opportunity?
For me, and I'll just a couple comments here. I think there's a real opportunity in multiple cancers, and I think we're seeing some sort of pan cancer targets that are very exciting, and I think we're also seeing targets that are associated, not necessarily with the cancer cells, but with other cells that are infiltrating the tumor, such as, fibroblast activation protein, which is showing a lot of exciting results with multiple trials out there. I don't know if Kent has more.
Well, it's interesting. Novartis put out a chart in the last few weeks that's showing essentially a migration of ADC targets to the RLT setting. And I think that speaks to why there's a high level of interest in this field, and that is it's potentially complementary to existing treatments because it's a different mechanism of action. And so you can have a drug that's, say, right now, or second line as a warning, black box warnings, all of which are driven by the immune system. And then you have this treatment which doesn't use the immune system, and so you can potentially have a cleaner, at least go in and reduce the tumors incrementally on top without all the same side effects.
Yeah, that's interesting. And it's also interesting to mention these antibody drug complex. I think that has opened the door for obviously targets that are good tumor targets, but you have the added benefit of maybe relooking at some of these other antibody drug complexes that weren't maybe so successful, but in this context, you don't have to worry about the targeting agent being taken up in the cell, because you just have to get the radiation dose close enough, and you can have bystander effects where you can have also nearby cells that don't even have those tumor targets on them. So yeah, so that's kind of like where I was asking the questions, like what makes a good target in the first place, as far as
Very, very high levels of expression on the tumor and nowhere else.
yeah, for sure.
And one example to look at that's not looking, hasn't worked out so well recently is J&J's 6420, where there was data presented at ASCO. Now this is targeting HK2 in prostate cancer. HK2 looks appealing because of its high correlation with PSA. Now it's using actinium, which is a high energy isotope, but it's I don't recall the exact numbers, but it's not as highly expressed as PSMA. And the initial challenge they're encountering in the trial is that the actinium gets to the target, but it's going a lot of other places, and they're still pursuing it, but the tolerability is a challenge here. And in fact, the KOL running the trial said, it may be that this is a target that's more appealing for a non-RLT therapy.
I think one thing too to consider with actinium-225, and you kind of alluded to that before, is the daughters. And so actinium-225, instead of just being this nice decay that releases one alpha, it decay. And so there are radioactive species that are released, and in most cases, they will be released from the chelator, and they can be migrate a little ways from the body. How much that happens is likely very dependent on internalization of the targeting vector, perfusion in the tumor, and other aspects. And I think that those kinds of things need to be studied as well, so that we can better understand a lot of the off-target toxicity, because I think that at least some of the off-target toxicity is likely because of those daughters.
Yeah, certainly. And then that goes back to like the designing of the chelators, and linkers. And is there a possibility to design like a chelator that could handle all of the daughters as well as the initial isotope? That's
so we should be careful about expectations. There's likely not a chelator that can handle an alpha decay and stay together.
right.
I think that would be, like the traditional chelator. Now people have made, nanoparticles and different kind of multi-scale things, or different kinds of complexes that people have looked at. But, the idea that a traditional chelator could handle an alpha decay, I think as a chemist, I find that a little unrealistic.
Yeah.
It's possible with some of electron capture decays. So there are other isotopes that decay in multiple ways, that in a fashion that could be contained by a chelator. But I think understanding the distribution of those daughters, and there also may be secondary measures that you could use to essentially capture the daughters and get them out of the body in a rapid fashion.
Sure.
There are other isotopes such as, astatine-211, that only decay via one alpha emission. But then, of course, the chemistry is totally different again, and then we're redesigning things. So I still think there's a lot of chemistry and biochemistry work to be done in this area so you can really move the needle on how to move these forward most effectively.
I've been observing some chelator technology just entering the clinic that's really intended to increase affinity.
Mhm.
So that you can use lower doses of the isotope, in this case, actinium-225, to address, well, it addresses supply chain issues, but then also addresses the safety issues. So we'll see how we'll get a sense of those over the next, I think, 6 to 12 months in one or some cases.
Sure.
Absolutely too. And I think that this is also really coupled to the targeting vector. So in some cases, if you're using a targeting vector like an intact antibody, because you're pulling that knowledge from the ADC literature, that antibody circulates in the blood for a long period of time. And so, it has there are some decays that are going to happen while it's still in the blood, and therefore you're going to have some decays that happen that are not giving you therapeutic efficacy. Whereas some targeting vectors that may localize to the tumor very fast, like some of the PSMA agents that are out there, may prevent that blood circulation. Now, on the other hand, those small molecules, they clear through the kidney and a lot of the drug is excreted. And so then you have different toxicity profiles. And so I could see, this is why people are interested in like cocktails of isotopes or cocktails of targeting vectors that may be useful, or combination therapies where they can kind of stratify those toxicity profiles so that they're not all hitting one area.
Yeah, certainly. And then, when you're talking about toxicity and alphas too, I mean, since they're so powerful, you can get it down to just a few alpha particles to destroy a cancer cell. So if you can get these high affinity targeting elements with alpha, I mean, those are gonna work together for that. But yeah, you're correct too, but this high energy also makes it a challenge to make sure that you're keeping it tethered to that targeting agent to get it to that target before you get too many decays in the blood. So certainly another area that requires attention, and it's not just it's not just the radiotherapeutic developers that need funding and attention right now, but it's also these supporting chemistries and supporting the supporting providers, at the patient end. There's a lot of buildup that we still need to make this a widespread success for radiotherapeutics. So let's see, but let's think a little more general. I mean, there were two big meetings we just had, both mentioned recently. We had ASCO and SNMMI. So maybe both of you, to close this out, could talk about what you thought coming out of those meetings were the biggest takeaway as far as the near-term future for radiotherapeutics and diagnostics, if any.
So I attended SNMMI, and it was the energy around this field was really great, and the enthusiasm and I want to say curiosity for new isotopes, what's next in the pipeline, what's next in the toolbox, and how we can move these things forward was really exciting. A lot of really early phase clinical trials, and a lot of companies moving forward in this space. So I feel like there was developments from the isotope production, from chemistry to clinical trials and moving things forward. I think that's also coupled with exciting advances in new scanner technology. So getting better images of the diagnostics to help us understand how we can use the therapeutics, things like whole body PET-CT, improvements in higher sensitivity SPECT scanners it's all kind of working together to move the field forward. So I felt like at least SNMMI was a very positive meeting and really showed a lot of energy and enthusiasm for moving a lot of different technologies forward.
How about you, Kemp?
I would concur with that. I think of the industry right now as being a giant dose ranging trial. And it's, new targets, new isotopes, new linker technology. And building onto that, I met with a number of different companies, and I can think of about 4 or 5 companies that are actively in the market looking for capital, which is near and dear to our hearts, that, I met with. So it's there's a lot, it's very dynamic right now. And I think the other thing that I found interesting is, a quote that was relayed from one of the senior RLT people at Novartis, and that is, we've probably only tapped about, I think it's 7% of the potential targets.
Yeah, I'd agree. That's exciting. That's why we just need to build this infrastructure around it to get that toolbox of targets and linkers and chelators and isotopes and imaging and think that we can't even really comprehend the effect once we get all of this all of these tools in the toolbox for us in the future, because this MOI it's tried and true, right? We know radiation kills, and making the precision medicine aspect of bringing it to the tumor. I'm excited about it, and I'm glad to, have this discussion with you. I think this is a great starting off point. There's a lot of tangents that we can go off of and discuss certainly in more detail down the road, but I really enjoyed our discussion today. And I think with that, maybe if you have any closing words aside from that, other than that, well, let's move on to our questions from the audience. You guys good? All right. Let me just put my glasses on here, everyone. Give me a moment. Okay. Some of these we already touched on. So someone's asking here, what are the current clinical trials involving radioactive isotopes?
There's a long list.
It's a long list.
Yeah. I think that there's a lot of clinical trials involving radiopharmaceuticals that are already approved, kind of moving them into new indications. And then, there's a push to move a lot of these alpha particles forward. I don't know if, Kemp, you want to talk about the landscape a little more?
It's there's no simple way to find them all. I mean, just when we go looking for trials that haven't been announced by a developer, you go on clinicaltrials.gov, and you have to just go by. I just go by isotope, what shows up, and do it that way. And it has exploded over the last 2 years since we started publishing the Roundup. So it's and the list is growing.
Yeah, certainly, which is good. That makes me happy to see that. So let's see, somebody asked actually a more basic question here, which is, what is a Curie? And how does one calculate how much energy and cytotoxic effect is distributed to the tumor by a radiopharma drug?
Yeah, that's a great question. Okay, so a Curie is a measure of how many decays per second. And so a Curie is a more historical reference. It actually refers to the amount of radioactivity, I believe, in a gram of radium. And so it equals, let's say, 3.7 times 10 to the 10 decays per second. So it's quantified in the number of atoms decaying in your sample per second. So traditional radiotherapy does calculations of the amount of energy deposited in the tumor. So when you have external beam radiation, they will say so many gray, which is energy per unit mass. And so we can try to do some of those same sorts of calculations with, we know we have this many atoms decaying, they're releasing this much energy. And then we can get an idea of how much energy is decaying or is being distributed per cell or per tumor or per gram as you can do those calculations. There's a lot of medical physics work underway, because we're realizing that actually where that energy is distributed, whether it's distributed in the cell nucleus or the cytoplasm or outside the cell or near the cell, that can really impact the cell's response. And so that's sort of the basic underlying idea. But I think understanding the differences between traditional external beam radiation electron emission or beta decay, alpha decay, and then maybe eventually Auger electron decay we're still trying to understand that. And there are still a lot of great people working in the medical physics realm and trying to understand how cells respond differently to all of those. The other thing is that different cells respond differently to different amounts of radiation. So some types of cells are more resistant, some cells may be more susceptible to radiation, and we can do our best to understand which cells they are and understand, biomarkers to help us understand how we can maybe sensitize cells to radiation and make our radiation more effective. And so I know that's kind of a long-winded answer, but it's a great question because it's a question lots of scientists are still working on.
And actually, on that note, someone else asked, can you explain what dosimetry is and with a little more detail?
Yeah, so dosimetry is understanding how much radiation goes to each organ. And so basically, if we give you a radiopharmaceutical, we would love it if all the radiopharmaceutical just went to the tumor. But the radiopharmaceutical will also be excreted through the body. And so you will have some radiopharmaceutical that ends up in the liver, some that ends up in the kidneys, or depending on the molecule and how your body gets rid of it. And so dosimetry is understanding how much radiation dose goes to each component of the body and goes to the tumor, goes to the liver, goes to the kidney. And so the idea is to optimize and target the dose to the tumor so that we get a high enough radiation dose to the tumor versus the other organs. We can calculate dosimetry by using these imaging isotopes. And so the imaging isotopes won't give you this high radiation dose that the therapeutics give you. So we can use the images also to predict what the dosimetry will be from the targeted radiotherapeutics. And that is also sort of a more precise way to use dosimetry. But dosimetry on a whole is really just figuring out how much dose to each area of the body.
Right. And I think one thing, when I'm thinking maybe from a more layman's standpoint, when you're talking about radioactive isotopes and some are for imaging and some are for not, and you're talking about energy, I mean, I think of it as like when you're doing imaging, your energy is like you're taking a picture, right?
Mm-hmm.
And so that's not going to be as detrimental, but the higher energy, it has more energy to like blast apart a tumor cell. I don't know. That's a good way to
Yeah, and higher localized energy. So you can
localized energy.
The imaging isotopes emit radiation that's emitted from the body so that we can see it.
Right.
And we can see it using those cameras or scanners. The therapeutic isotopes emit radiation in very short path lengths. And so that energy or that radiation is specifically delivered to this very small area of the body.
Okay, let me see. And then one more on that. I'm going to say, somebody asked, can you talk about the advantages of Auger emitters versus alpha?
Yeah, so alphas, I mean, as we said, they distribute a lot of energy in a very small area. Auger emitters are very interesting because they emit a very high amount of energy in an even smaller area. So the Auger emitters don't even go as far as a cell. And so the idea is if you can get Auger emitters specifically to the cell nucleus, you can deliver radiation on a per cell basis, on a very, very small basis. Now, the chemistry with Auger emitters and the chemistry to deliver those isotopes to the cells is still definitely something that's ongoing and something a lot of people are interested in. But the idea is that you can deliver radiation in an even more localized fashion. But of course, that's very, very hard to do and hasn't yet been shown. The efficacy, at least in animals in clinical trials, has yet to be sort of demonstrated. But I believe that there's been a lot of progress on making the isotopes and the chemistry. Now we just have to figure out we're even going one step further. We've been figuring out trying to get isotopes delivered to cancer cells. Now we're trying to deliver it to cancer cell nucleus, which is even more difficult.
Okay, I'm going to switch gears a little bit. And someone asked the question, can someone comment on what the cost burden to the patient is for these developing treatments and therapies, and how do they compare to the current standards of care?
Right. Well, let's focus on therapeutics first. So for Medicare, there's probably 20% copay. So as I mentioned earlier, at around $50,000 per dose, the copay could be up in that ballpark. And again, that's without dealing with the specifics of Part D coverage. Now, that pricing is, if you think of a course of therapy, it's in the range of what we've seen in other innovative oncology therapies. So, I think that was by design, keep it in that, keep it in that range. The diagnostics, they are at a premium to the older therapies, older options such as FDG. But again, $5,000 per dose, so you are potentially looking at up to $1,000 out of pocket, at least with Medicare. So, those are the ballpark numbers.
I see. Sorry, we have so many questions, everyone. Excuse me, I'm just trying to get through them. I'd love this. This is great. So someone asked also, what is the market potential for radiopharmaceuticals, since we're on the finance end? What do you think?
Yeah. I haven't, personally, I don't have a large, a big number out there to work with. There are, various third parties that have come up with estimates in the tens of billions of dollars. And again, you think of some of the products that are already out there. So Pluvicto is annualizing at north of $1 billion. Lutathera had been around 600-ish million, but that's going to increase pretty sharply because they've just shown clinical data showing use in first line, and they don't have to go back to the FDA for that. They can just start promoting that. And then, some of these, in Pluvicto, we're talking about potentially going in the pre-taxane line of therapy, and that'll, I think, roughly double the target market. So, again, think of the math of, around $200,000 per course, for a full course of therapy, and on tens of thousands of patients in a given indication, and you can come up with some very large numbers in the tens of billions of dollars pretty easily.
Okay, so another person asked, what are the key challenges to consider for clinical trial design, including enrollment for radiotherapeutics?
I think
I can go ahead, Susie.
Yeah, no, why don't you go ahead first?
Okay, I will say this: we've seen with prostate, and this was initially with the Vision trial in Pluvicto. So, typical trial design in that there's a control arm and a treatment arm, and initially they had trouble enrolling because patients could figure out if they were in the treatment arm or the standard of care arm. So Novartis amended the protocol to allow patients on the control arm to, if they progressed on standard of care, to then cross over to the treatment arm. And I think they had about 80% of the patients on the treatment arm ultimately cross over. So this had the effect of confounding OS calculations. And Pluvicto got its first approval without a survival claim. It was just for rPFS. Now, as a result, with PSMA-4, they've had to do the same thing, and that has led to a delay in filing the supplemental NDA because FDA wanted to see the survival data mature, and it's, I think, at a point where the hazard ratio is less than 1, and Novartis is proceeding with the filing. Splash trial, which is lutetium-177 based radiopharmaceutical for same indication as PSMA-4, and then Fusion's. I want to get the trial Alpha Break trial. When they go into phase 3, they're going to have that same crossover design. And and as a recognition by clinicians, you need to have that crossover to get enrollment. And so that's a challenge with trial design. You don't get the clean data at the first pass. You have to wait.
And let me see, I want to go, sorry, I don't think we're unfortunately going to be able to get to all these questions, but I know we have some about production. Maybe I can ask one or two of those before we're at time right now, but we have so many questions. So it says, can someone comment on the challenges facing novel actinium-225 production technologies such as radium-226 supply, radon-222, and target cooling, etc.?
Yeah, I think those are definitely all considerations. I think getting radium-226 and having a good supply of radium-226. So actually, let me back up. For those of you that aren't familiar, several of the methods to make actinium-225 require the use of a radioactive target. And so instead of just taking a target material and bombarding it with protons or neutrons or something, now we have a long-lived radioactive target, and we're bombarding it with protons or neutrons or gamma rays to induce a nuclear reaction. Then we are separating our newly produced radioactive material from the previous radioactive material. So the challenges associated with that, I think, are clear. We have to contain, the target containment is much more of an issue than it is when we're working with stable isotopes. And the target material is decaying, and so you're losing it, but it's decaying into radon, which is a gas. And so the other thing is that now we have a radioactive gas to deal with. The technologies that are being developed are very interesting. I think that a number of different groups have addressed them in different ways, but I don't think that there's really a completely solved system that's generating lots and lots of actinium for all these clinical trials yet. We are starting to see actinium come from some of these novel these novel production methods, which is really exciting, because I'm very glad that the chemistry and the physics and engineering involved with these targets is moving forward. So I think we'll see a number of companies moving this along in the next several years.
Yeah, just to add with actinium-225, so the Department of Energy's, and I think it's their accelerator version of actinium-225, yields small amounts of actinium-227, which has a half-life of over a century. I think that's right, Susie. And as a result, there are additional disposal and bonding requirements for its disposal that even though for clinical trial purposes, FDA is comfortable with the level of 227 that they're producing, the trial sites just don't want to deal with it, except in the smallest quantities, and so that's a real constraint with that isotope.
So actinium-227 has a half-life of 20 years, which, in the general scheme of things is still very, very long. And that is the production by very high energy protons on thorium targets. So it's a little bit different than the radium production pathways. And in fact, that 227 issue is why people are looking at the radium-226 pathways that could give much cleaner actinium-225.
Okay, I think I'm going to fit in just one more question here to wrap it up, because we're talking about building a toolkit of all different combinations for radiopharmaceuticals. So what do both of you think would be the next exciting tool that we need in the toolkit to really advance radiopharmaceuticals? I know we mentioned chelators, maybe.
Yeah, I think, it's hard for me to pick one, because the isotope development really pairs with the chelator development, pairs with the targeted development. And I think if we can really get an understanding of, how to better pair isotopes to chelators to targeting vectors, and make them so that we have a better idea of understanding of target uptake, but also a better understanding of how those all those factors impact clearance and excretion and off-target uptake, will be better equipped. And so I think more people working on multiple things simultaneously would be my non-answer to your question.
Well, my answer is going to be the linker technology, because it's potentially the most elegant.
Yeah.
And addresses both safety, but then also supply constraints.
Great. Well, thank you for everybody's time and attention here. I think the fact that we have so many unanswered questions, all I can say is that I think it offers a good jumping off point for further discussions that I hope we're going to have in this regularly in this area. So thank you, everyone, for your questions and participation, and have a wonderful rest of your Friday.